Hi friends, in this we are going to discuss about the difference between epistasis and dominance. First we need to know the definition of epistasis and dominance. Epistasis, it refers to interaction of two or more genes, thus it involves two or more loci. That means it between interaction between two or more genes, two or more. But in this dominance, it refers to interaction between two alleles of the same gene. Thus, it involves single locus. In epistasis, interaction between two or more, like suppose these are four genes, interaction between all these interaction happens between all these genes. It's called as epistasis. But in respect to dominance, these two are only single gene, but two alleles, capital A or small a, like this. These are the alleles, capital A, small a. Interaction between these two occurs. And this is for dominance and this is for epistasis. It may involve both homo and heterozygotes, hence it is fixable in homozygotes. But with respect to dominance, it is always refers to heterozygotes and it's not fixable, because in case of dominance we need heterozygote. Only dominant will only express and the recessive one will be suppressed. So it has to suppress something that it is in recessive condition. So one should be dominant and one should be recessive, so that dominance can work. With respect to epistasis, it's interaction between, it can occur homozygotes like capital A, B. Suppose these two genes are interaction between these two genes can occur. Suppose this code for a particular gene, interaction between these two can occur. So like it's interaction between like this capital A and small c, capital C, small c. Interaction between all three genes can happen. Suppose in homozygotes we can fix in homozygotes and it's not fixable. That means whatever the epistasis, if the favorable interaction is present, if the favorable interaction will occur and we can fix that, that means we can transfer from one progeny to other progeny, from one generation next generation after reproduction. But it is not with the case of dominance, because continuous segregation will occur, so it's not fixable with respect to non-heterozygotes. So heterosis can be exploited. Epistasis is of several types such as dominant, recessive and duplicate. But with respect to dominance, incomplete dominance, complete and over dominance can occur. These are types of dominance. Incomplete means like as we see in red, pink and white flowers, it's called as incomplete dominance, complete dominance and over dominance. Suppose full complete darker red color or darker green color occurs, then the parents, and it modifies, and with respect to epistasis, it modifies normal dihybrid phenotypic ratio in F2 and a partial dominance alters the normal segregation ratio 3:1 into 1:2:1, as I said. Partial dominance or incomplete dominance, I said. It also known for intergenic interaction. Intergenic means between two or more genes. Intergenic or interlocus. But it is all intragenic. Between one gene there will be two alleles. The interaction occurs between two alleles, and intralocus, within that locus only the interaction can occur. Recessive gene can also exhibit masking effect. And recessive gene, in this dominance, recessive gene can express only in hemizygous condition. After we studying the similarities, that is difference, we can move on to similarities. It also affects same characters. It also affects particular characters. This is nuclear genes. This is also non-nuclear genes.